Hi, my name is Blonid McKeown and I'll be describing how the bases are attached to their sugar phosphate backbone. As Molly said, DNA is composed of nucleotides, and nucleotides have three elements. The first being a sugar, second a phosphate, and the third is the base. The sugar is described as a monosaccharide, that is deoxyribose. Deoxy meaning there's no hydroxyl attached to it. And extending from the anomeric carbon is a heterocyclic base. It is a heterocycle as it is a ring structure with at least two other elements as members of the ring. In our case, it's nitrogen and carbon. It is also a base because the nitrogen atoms have a lone pair. I'll describe this further in the next slide. These bases are attached to our anomeric carbon by a beta glycosylic link. As you can see, it is attached to our carbon 1. Firstly, the bases that are found in our DNA are adenine, thymine, guanine, and cytosine. They are often noted as A, T, G, and C for simplicity reasons. The bases are attached to the first carbon of the sugar ring by the glycosylic linkage that we showed in the previous slide. All the bases that are found in DNA contain nitrogen atoms, and they are all cyclic structures. Our bases are subgrouped into purines and pyrimidines. Purines include adenine and guanine, and these are double ring structures. Pyrimidines include thymine and cytosine, which, as you can see, are single ring structures. Complementary base pairs always involves one purine and one pyrimidine base. Adenine, a purine, bonds with thymine, a pyrimidine. Two hydrogen bonds are formed. A hydrogen bond is a weak bond between two molecules, resulting in an electrostatic attraction between a proton in one molecule and an electronegative atom in another. In this case, the hydrogen on adenine is the proton donor to the oxygen in thymine, which is a proton acceptor. And for the second bond, the nitrogen in adenine is the proton acceptor to the hydrogen in thymine, which is a proton donor. And next, guanine binds with cytosine, but this time we have three hydrogen bonds. The oxygen from guanine is the proton acceptor, and it accepts the proton from cytosine from the first bond. The second one, guanine supplies the proton for the hydrogen bond to the nitrogen, which is the acceptor. And the last bond, guanine supplies the third hydrogen to the oxygen and cytosine to accept that proton to form the third hydrogen bond.
As mentioned earlier, DNA is double-stranded. The two strands are connected with non-covalent hydrogen bonds by the joining of base pairs. Base pairing is highly specific. One purine and one pyrimidine fit perfectly together between the two strands. Everywhere there is a cytosine on one strand, we have a guanine on the other strand. And everywhere we have an adenine on one strand, we have a thymine on the other strand. This specific pairing is down to the hydrogen bonding between the complementary bases. An adenine-thymine pair makes two hydrogen bonds, and a cytosine-guanine pair makes three hydrogen bonds. The two strands of DNA are antiparallel. Looking at the phosphate backbones we mentioned earlier, we see they are upside down in relation to each other. One runs from what is called the 5' to 3' direction, which means the first phosphate is connected to the sugar molecule's fifth carbon, and the next phosphate will go ending at the 3' end of the sugar. The complementary strand will then go from the 3' end to the 5' direction, where we get the exact opposite.